Welcome back to What I Did in Lab today, where I tell you guys what I did in my biochem lab today. And this is the last lab of the semester, so this will be the last of the series for a little bit. So today was SDS-PAGE, and if you've been around my account for a while, you know that I have beef with SDS-PAGE. It's a long story. So if you remember from last week, we had a bunch of protein that we purified and measured the absorbance of, and then we stored for use in this lab. So we were running the protein that we purified last week on our SDS gel, but first we had to do a couple things. So what we had our three samples, the soluble protein, the flow-through, and our actual purified protein, which we had to mix with some loading dye to be able to actually visualize and see on the gel. So what we did, we had to mix those up and leave them to incubate for a while in order to just get all of their stuff done, and while we were doing that, we had to set up our SDS-PAGE contraption. Now, I am not a big fan of SDS-PAGE. It is a pain in the ass and I hate it. But what we have here, this is our actual gel. That green thing in it is the comb that is there to separate the different wells, which is what our protein goes into. Then here in the clear we have our actual gel, which is sandwiched in between two thin pieces of plastic because the gel is very thin and has the structural integrity of wet spaghetti. So what we had to do was take out our gel from that package and load it into this big plastic apparatus. Now, there was actually two groups per gel, so we were sharing with another group. There's me and my lab partner, and the other group, which is my friend and his lab partner. So what we had to do after we took our gel out of the package, we had to put it into this plastic thing and very, very carefully take out this green comb so as not to destroy and rip the very delicate gel wells. And then we had to fill the entire thing with loading buffer, and then we had to kind of clean and rinse out the wells just to make sure they were all clean, which our first mistake of the lab came up. So now this wasn't me or my lab partner, it was one of the other guys in the group who, when he was cleaning out the wells, he thought that he broke one of them. He thought that the gel tore and one of the wells was all fucked up. It turned out to be okay. It turned out to not be a big deal, but that was fun. We did that while we were waiting for the proteins to incubate. When they were done, we very, very carefully, with a very long, thin pipette tip, got all of the proteins out and loaded them again very carefully into the wells because it is very, very easy to overflow the wells and just spew protein everywhere, and then you won't get a good result. So what we had our blank, our flow-through, our soluble protein, and our purified protein, and we also had what is called a ladder, which led us to our second fuck-up, which was on me this time, I will admit. So what happened, we had to have our ladder mixed with a couple other things. We had to mix it with some water and some loading dye, but I messed up the concentration, so it wasn't a big deal. It turned out fine, you couldn't even really tell, but I did mess up the concentrations on that a little bit. But it turned out fine. So we loaded all of our samples. I only loaded one of them because I hate doing it. It's stressful and it's very easy to fuck up, so I only loaded one. But loading actually went pretty well. None of the wells overflowed, none of the wells were punctured because, again, it is very thin, very delicate, and very easy to break. But fortunately, it went fine, so we stuck it in the buffer, we turned it on, and that led us to our third fuck up. So in the lab, there is technically two groups total. There is my group and my friend's group and the other group that we weren't involved with, but they forgot to peel off the strip off the bottom, and that reminded us that we had forgot to peel the strip off the bottom. So we had to very carefully disassemble the whole thing without, one, spilling buffer all over the lab bench, which is just a waste of stuff, and two, take out the entire plastic bit and very carefully hold it up and peel the strip off without spilling any of the protein, without tipping it, without dumping the buffer out, without getting the protein everywhere and like overflowing it into other wells and then putting it back in and turning it on again. That was stressful, but we prevailed and we got her done. So then after leaving it to run for a while, it was finally done, and then we had the stressful experience of removing the gel. So after it was all run and ready to be done, we had to crack open the two sides of the plastic, like a thin and delicate oyster, and then very, very carefully peel off one side of the plastic, leaving the gel stuck to the other side. Then we had to use this green thing and a container of water to very, very carefully peel the gel off of the other side of the plastic to get it into the water. Now, I got my lab partner to do this because I did not want to. He very, very carefully, it took like a solid 5 minutes, very carefully scraped the gel off of the other side, being careful not to tear it, because again, it's very delicate and very easy to tear. But he got it done fine, and we eventually got it into the container, where we then had to wash it 3 times, 5 minutes each, very time consuming with a bunch of water. Then after washing it a few times, we had to let it soak in the stain because the protein on here doesn't actually show up unless it's stained, so we had to leave that rocking in its container full of stain for 30 minutes, during which I worked on my lab assignment. My lab partner fell asleep, and our other 2 partners were poking him with a pencil and messing with his YouTube videos that he was kind of watching. Then, when it was finally stained, we had to wash it again. Then, finally, we got our results. So this, obviously, isn't the actual gel, it's an image taken of the gel. So what we have here, we have mine and my lab partners on this side, and then mirrored our other lab partners group on this side. So they're the same thing, they're just mirrored opposites. So what we have in the middle here is the ladder. As you can see, my mess up with the concentrations did not have any effect on how the ladder looks. This is just a mix of proteins of a known size to be able to compare our protein with to see how big our protein is. So how SDS-PAGE actually works is it works by size differentiation. So proteins of a larger size move through the gel a lot slower, so they stay more at the top, whereas smaller proteins move through quicker and go down the gel farther, so they're closer to the bottom. Then what we have over here in this is our blank. There's no protein in it, which is what we want. This is just a comparison, basically a negative control. Then these 2 in the middle, those are the soluble protein and the flow-through. As you can see, it's just kind of a bunch of big streaks with a couple distinct bands. This is just a bunch of junk protein that we don't want that got purified out during the purification process. Again, this is just for comparison to make sure that we actually got rid of all of the junk that we don't want. Then finally here we have our purified protein sample. Now, admittedly, it does look like there is nothing here, but on the actual physical image I have, it does show up a lot better than it does on this camera, but you can very, very faintly see about right here, there is a band. This is our purified protein. Now, since it has been purified so much, there is less stuff in it, so you don't really see it show up as much or as distinctly, but it is there and it is at relatively the size we want, so it does show that we purified the correct protein and we do have protein here. Then, finally, when we were done, we decided to rip it up into little pieces because we were mad at it because it's a pain in the ass. Now, I will say this is probably the best SDS gel I have ever run, which is not saying a lot. Obligatory moment of silence for the worst gel I've ever run. So I'm the only one actually out of the 4 of us that has done SDS-PAGE before, but now they know why I hate it so much and they kind of agree that it is a pain in the ass. But yeah, that was the finale to the lab.